Hello, my name is Molly Herron, and I'm going to be a junior at the University of Great Falls in Montana.
Hi, my name is Judy Yao, and I'm a rising sophomore at Tufts University. This summer, we've been working in Dr. James' Kaufman lab, and today we will be talking to you about the effects of chronic cortisol exposure on the innate immune response in zebrafish.
So what is stress? Stress is a reaction to a stimulus that offsets our body's equilibrium. There are two major types of stress, acute and chronic. Acute stress is often short-lived and contributes to our body's fight or flight response.
On the other hand, there's chronic stress. And chronic stress is when you're stressed out for a longer period of time. Chronic stress may contribute to many diseases later on in life. These health problems may include Alzheimer's, arthritis, cancer, cardiovascular disease, depression, diabetes, and other inflammatory diseases.
So how does our body respond to stress?
Our body responds to stress through the HPA axis. The HPA stands for hypothalamus pituitary adrenal axis. So when you're stressed out, a signal gets sent out to the hypothalamus, and that sends a signal down to your pituitary gland. And your pituitary gland releases a hormone that is interpreted by the adrenal gland. And your adrenal gland releases a stress hormone, which is a glucocorticoid called cortisol.
Once cortisol is released into the blood, it moves to the cytoplasm of the stress cell, where it then meets and binds with a glucocorticoid receptor. Once the cortisol and glucocorticoid receptor are binded, the glucocorticoid receptor acts as a transcription factor, which regulates genes that respond to stress. After the acute stress is resolved, the HPA axis is usually shut off through a negative feedback loop. However, when there's chronic stress, this negative feedback loop is unable to shut off the HPA axis. This causes a constant release of cortisol, which basally increases the level of cortisol throughout the body. Recently, the Kaufman lab has published a paper on how cortisol exposure affects zebrafish. And what they found is that larvae treated with cortisol had an upregulation of genes associated with defense response and immune system processes. They also found that adults derived from these larvae had an abnormal regulation of genes associated with defense response and immune system processes.
A recent study done by a research group in the Netherlands acutely exposed 3-day post-fertilization zebrafish to the synthetic glucocorticoid called beclomethasone. And they found that this chemical inhibits neutrophil migration. It has no effect on macrophage migration, and it inhibits pro-inflammatory gene regulation.
Combining the research method of the group in the Netherlands and the interests of the Kaufman lab helped us come up with our research question, which is, how does chronic cortisol exposure affect the innate immune response in larval zebrafish?
Our hypothesis was the cortisol-treated zebrafish will not affect macrophage response, but it will have an inhibitory effect on neutrophil response to a wound. And this summer, we studied the migration patterns of neutrophil and macrophages. And this is a time-lapse video of immunocytes going towards the wound site.
Neutrophils and macrophages are both part of the innate immune response, which is the defense mechanism that comes into immediate play once an antigen is detected.
So what are neutrophils and macrophages? Neutrophils are the first ones to respond. They're the first to fight off any invaders, and they are the ones that signal macrophages to come to the wound site. And macrophages are the ones who engulf pathogens, dead cells, and tissue debris.
The reason we chose to look at macrophages and neutrophils is because their initial myelopoiesis, or formation, occurs between 12 to 19 hours after fertilization. For our model organism, we chose the zebrafish for 3 key reasons. It has the capacity to regenerate a tail fin, its immune system is similar to that of mammals, and you have the option of using transgenic lines.
The 2 transgenic lines that we used this summer were MPX and MPEG. MPX has a GFP, or green fluorescent protein, in its neutrophils. The MPEG line has a YFP, or yellow fluorescent protein, in its macrophages.
After we bred these two transgenic lines, we collected their embryos and placed them into treatment immediately after fertilization. Our control group was treated with DMSO vehicle, while our treatment group was treated with 1 micromolar concentration of cortisol in order to elucidate the effects of chronic stress. Both these groups were kept in their treatments until 3 days post-fertilization. At 2 days post-fertilization, larvae were screened for their fluorescent markers, and at 3 days post-fertilization, larvae were used for tail fin amputation, time point imaging, whole tail fin imaging, or qPCR to measure gene expression.
So when we were doing time point imaging, we imaged the fish at 6 different time points. These were at 0, 2, 4, 7, 24, and 48 hours post-amputation, or HPA. So in the MPX line, we see that the treated fish have fewer neutrophils than the control. However, our results were statistically insignificant.
In the macrophage line, we found that the treated larvae actually had a more number of macrophages moved to the tail fin amputation site compared to the control larvae. However, these results were also statistically insignificant.
After seeing that there were fewer neutrophils and more macrophages in the treated fish, we decided to dig a little deeper. So we decided to see if cortisol exposure would have an effect on the response on the leukocytes, the immunocytes, or the number of macrophages and neutrophils in the larvae. So when we're looking at the MPX line, the data for the whole tail fin, we didn't see anything statistically significant between the two treatment groups.
However, when we looked at the macrophage line, we consistently saw that the treated fish had a larger number of macrophages found throughout their whole tail fin. And to get a better idea of whether there was more neutrophils and macrophages in the treated versus the control fish, we decided to do qPCR to measure gene expression.
So we used qPCR to measure the gene expression of MPX and GFP. And we expected a similar fold change between the expression of these two genes because the expression of GFP is controlled by the MPX promoter. However, when we look at these graphs, we didn't see anything statistically significant.
In the macrophage line, we measured the MPEG and YFP genes. The MPEG is an endogenous gene, and the YFP gene is a transgenic gene, and they are both found inside macrophages. And what we found is both of these genes had a 2-fold increase in the treated larvae. This means that both genes were upregulated in treated larvae. Our results brought us to these conclusions.
So our conclusions are the number of neutrophils at the amputation site and throughout the whole body are not significantly affected by the cortisol exposure throughout development. We also found that chronic cortisol exposure throughout development increased the number of macrophages at the amputation site and throughout the whole body.
This evidence suggests that chronically elevated glucocorticoid signaling specifically upregulates myelopoiesis or formation of macrophages. Our acknowledgements. We would like to thank the Kaufman Lab.
We would like to thank our PI, Dr. Kaufman, our research assistant, Ellie, Sushan, and Ian. I would like to also thank my funding source, the James Slater Murphy MD Fund.
And I would like to thank my funding source, the National Science Foundation REU program. And we would both like to thank the MDI Biological Laboratory for allowing us to spend our summer here.
Thank you for your time, and we'll take any questions now.
Questions for Molly and Judy?
Very nice. Do you know where these cells come from? These white blood cells in the fish? In humans, they are made in bone marrow. Where do they come from?
I think they're made in bone marrow too. I don't really know the answer to that question.
The question was that where are these white blood cells formed in the zebrafish? And
We should probably know that.
we're not
We
really
do
sure
not.
about that.
We don't know this for sure, but we actually read a paper yesterday, and it could have something to do with an IRF8 gene, which is involved in the myelopoiesis of macrophages, but that's just recent evidence that just came out. Yes?
Why did you choose to look at larval zebrafish as opposed to adult zebrafish?
Ellie and Dr. Kaufman looked at adults, and they found some evidence in larvae, and they just wanted us to take a look at how it affected the innate immune response in larvae.
So the term chronic can be a challenging term to deal with, right? Because if you think of medicine, acute typically means days of time to see things, but hyperacute would be hours, and then chronic can be decades. Like if you give a transplant, it could be rejected acutely or chronically. And so you're using the term chronic exposure in embryos that are developing for a short period of time. I'm curious how you rationalize that as a chronic exposure.
So the question is, what does chronic mean to us in our research? And to us, it means that the fish are constantly growing in a media with cortisol. And other studies define acute as exposing these fish with cortisol or other synthetic glucocorticoids for a few hours, or like not the whole time while they're developing.
I have a question. I don't know if the results are from adult zebrafish or from embryonic zebrafish, but other results from the Kaufman lab show that chronic stress reduces, as I recall, reduces the ability to repair wounds and slows regeneration. But your results seem to show that, your results clearly show that macrophages increase in number. So what gives? It seems that if regeneration is slowed, but you see more immune cells, what might explain that?
The question is, we see more macrophages in our research, but our lab's previous research shows that regeneration is slowed down when they're adult fish. And how does that work? So our research showed that there are more macrophages, but we aren't really sure if the fins are like developing slowly or if they're developing like faster, because we haven't really looked into that. But that's a good idea for the next project.
Other questions? Thank you very much, Molly and Judy.